And Sebastian, you have the invite. Oh yeah, there you go. So just a quick introduction. Some of you know Sebastian, but Sebastian is the founder and CEO of PAIA Biotech, based in Cologne, Germany. And he has some novel assays for quantifying characteristics of antibodies. And today he is going to talk about some glycan properties, including afucosylation. And so I'm going to turn it over to Sebastian.
Thanks, Chris, and welcome to this webinar, everyone. Great that you can take the time today to listen to my talk about afucosylation. So this is going to be the focus of my talk, and how we use our special kind of assay technology to provide a means to screen early and very quickly a lot of samples in cell line development. So I would like to start with a very short introduction to those who don't know PAIA yet. So quick introduction on PAIA Biotech. As Chris mentioned already, we're a German company based in Cologne, and we do have a special kind of a platform technology that we've been patenting, and that allows us to simplify, to streamline bead-based assays. And with this kind of assay technology, which is centered around some quite fancy microplates, we do two main things. First is we have developed high throughput titer assays. Actually, a new version for the FC titer assay was launched last month, which is extremely fast and really nice a new assay system. I'm not going to talk about that today, but we're going to focus on the assay kits that we've developed for glycan screenings of antibodies and FC fusions. So that not only means fucosylation, but also includes other features like galactosylation, mannosylation, Man5, big topic for antibodies, and of course acylation, which is the most important feature if you're looking at FC fusion proteins. But again, focus will be afucosylation today, and we do have two partners that we work very closely with. The one is on the technology side, which is Genentech, a company which is also a German company, and they develop and market very nice cell imaging systems with fluorescence, which we need for reading our assays. And we're working in the US with Chris and his team from Tek-Matic. They're based in Illinois. So that much as an introduction. So let's get to afucosylation. And firstly, I quickly wanted to remind us why we do all this. And the reason is because afucosylation has a very strong impact on ADCC. And this sketch here shows you how this antibody-dependent cellular cytotoxicity works. So the therapeutic IgG is recognizing the target cell, often that's a cancer cell, binds to it, and then recruits by its FC part natural killer cell, NK cells, which then do their job and kill the target cell. That's a very common mode of action for drugs against cancer indications. And this effect can be strongly influenced by the fucosylation on the FC part of the IgG. And this was first published in 2002 and 2003 by two separate groups from Genentech and from Kyoahako in Japan. And it took, precisely 10 years until the first drug, which was afucosylated, was approved in 2012 in Japan. And since then, we've seen really an increasing number of clinical trials involving those afucosylated IgG mainly in cancer, and that's what we also see at PAIA, that there's an increasing need and demand for screenings for afucosylated antibodies. So when people characterize the different glycoforms, especially also the afucosylated glycoforms, in the context of ADCC, they're mainly looking at two different kinds of assays. One is the Fc gamma receptor 3A binding, which can be done, for example, in our SPR systems, so surface plasmon resonance. That's what you see here on the left-hand side, multiple injections of increasing amount of different very well-defined glycoforms. And you see that the different glycoforms bind with a different affinity towards those receptors. I don't want to go too much into detail here, but the lower the degree of fucosylation, the higher the receptor binding becomes. And in this particular study, it was suggested that also galactosylation has a certain effect on this receptor binding. And then if the galactosylation is also kept with silylation, this affinity is diminished to a certain extent. So that's the one part or kind of functional assays that one does very often when it comes to testing these different glycoforms. And on the right-hand side, you see a typical ADCC assay, where we're looking at the response of NK cells to antibodies, and these curves here show the effect of differently afucosylated antibodies. On the left-hand side is the IgG with the lowest amount of fucosylation. On the right-hand side is 100 fucosylated antibody, which is usually the case if you don't do any kind of special kind of engineering to your CHO cell line, for example. So that's a huge effect that you can obtain by reducing the amount of fucosylation. And what is interesting in this plot also is that, for example, this blue line is something like 20% remaining fucosylation, so 80% afucosylation. So you can already get quite a big effect on the potency of the drug, even if there's a significant amount of fucosylation left on the antibody. So this is the one thing that I want to highlight here. The other thing is that it's not only the natural killer cells which have an impact on the ADCC, but there are also other cells which play a role there. Polymononuclear cells are involved there, and they also tend to prefer fucosylated IgG. So it's not necessarily a complete to get the best ADCC. So that really suggests that it's desirable to be able to tune the fucosylation to a certain extent in your production process. So that much for afucosylation and ADCC. And let's look at how people produce afucosylated antibodies and what are the different approaches to get there. So what people do, of course, is cell engineering, and that can involve genetic engineering, or the addition of special supplements to the cell culture, or a combination of both. And that can either lead to complete or to partial afucosylation of the antibody product. And kind of the most straightforward approach to get there is by knocking out the fucosyl transferase here in the middle, which means that all the glycans which are formed usually in this pathway and which are normally fucosylated become afucosylated. So with this kind of method, you get to very, very low degrees of fucosylation with not a lot of ways of fine-tuning that. You can also knock out genes further upstream this process, but this is not so common. So the knocking out the fucosyl transferase here is the original approach that Kyowa was pursuing early on. Then we have other ways. For example, that's what Roche Glycat is doing, overexpressing of these two enzymes to divert this pathway into this direction, which then leads to a production of bisecting glycans to quite a high degree, like 60–70%, and all lacking fucose. So these are two of the genetic engineering approaches. Another one would be, and that's what's shown here on the top, is knockout of these genes here, which kind of inhibits the cellular synthesis of fucose precursors here, so that the afucosylation cannot be done by the cell itself, but one has to feed in fucose from the cell culture media, and that is a means to kind of fine-tune or modulate the degree of fucosylation in the final product. So I think those are the most important methods for obtaining fucosylated antibodies at the moment.